These properties could be interrelated, might need to be considered in combination. To evaluate the potential effect of drug substance specific chemical properties on the performance of drug products, studies on drug product might be warranted. For example, ICSQ-6A specification test product seizures acceptance criteria for new drug substances, new drug products. Chemical substance describes some of the circumstances in which drug product studies are recommended. This approach complies equally to the Access Q6B, that is specification, test procedure, acceptance criteria, while biotechnology, biological products. The knowledge gained from the study's investigation, the potential effect of drug substance properties on drug product perform can be used as appropriate to justify elements of drug substance specification. That is 3.2 S 4.5. The compatibility of drug substance with other excipients listed in 3.2 should be evaluated. The compatibility studies with the excipient and API should be studied. For a product contain more than one API, the compatibility with the other API should be evaluated. Then second point 2.1.2 excipient. The excipient chooses their concentration based on IIG guidelines. The characteristics that can influence the drug product performance, that is stability, bioavailability, then manufacturability should be discussed related to the respective function of each excipient. This should include all substances used in the manufacturing of drug product, whether they appear in finished product or not. That is the processing rates. The compatibility of excipients with other excipients where relevant. For example, combination of preservative in dual preservative system should be established. The ability of excipient, that is a different category on, that is like a antioxidant, penetration enhancer, disintegrants, then release control agents for extended doses for extended dosing to provide their intended functionality. To perform throughout the intended drug product shelf life should be demonstrated. The information of excipient performance can be used as appropriate to justify the choice and quality attributes of the excipient to support the justification of drug product specification. Information to support the safety of excipient where appropriate should be cross reference. 2.2 drug product. The formulation 2.21 formulation development. The summary should be provided describing development of the formulation, including identification of those attributes that are critical to quality of drug product. The summary should be provided including the attributes of quality of drug product taken into consideration, intended uses, route of administration, which are considered uses of the dosage form, then route of administration, the dosage form should be provided. Information from former experimental designs can be used in identify critical or interacting variables that might be important to ensure quality of drug product. The summary should highlight the evolution of the formulation design from the concept up to the final design. The summary of product development from the initial development batches to the formal design should be incorporated. This summary should be taken into consideration the choice of drug product, that is the properties of drug substances, excipients, container closure, and relevant dosage device. The properties evaluated for the drug substance, then excipient, why excipients are chosen, then why container closure system based on the comparison with the RLD, then relevant dosage device, the manufacturing process, the if appropriate knowledge gained from the development of similar drug products. Any excipient ranges, including the batch formula, should be justified as a section of application. This justification can be based on experience gained from the development and manufacturing. Why excipients should be selected? Justification should be provided based on the knowledge gained from the development and manufacturing. The summary of formulation use the clinical safety efficacy in any relevant bioavailability bioequivalence studies should be provided. Any changes between the proposed commercial formulation and those formulations used in the pivotal clinical batches or primary stability batches should be clearly described in a rationale should be changes should be provided. The formula changes between the pivotal and commercial batches or the stability data provided should be changes in formula justification should be provided. Information from comparative in-vitro studies, that is resolution or comparative in-vivo studies by equivalence that links the clinical formulation proposed commercial. The comparison between the in vitro study, that is a dual resolution, and comparative in vivo study, that is back equivalent, that links the clinical formulation to be proposed commercial formulation should be summarized and cross reference to the studies should be provided where attempts have been made establish in a vitro and correlation. The results of those studies, cross reference of the studies should be provided in the section. A successful correlation that assists in the selection of appropriate dissolution acceptance criteria. Based on in vitro and in correlation study, we can establish the dissolution acceptance criteria. Can potentially reduce the need for further background studies following changes in the drug product or manufacturing process. Any special design features of the drug product, that is tablet core line, that is the break line on the tablet, overfill, anti-counterfeiting measures as it affects the drug product should be identified and rationally should be provided for their use. Then 2.2.2 overages. In general, use of overages of drug substance to counter compensate for degradation during the manufacturing product shelf life or to extend shelf life is discouraged. Overage added because of the losses can occur up to the shelf life of drug product till the product. That's why overage is added as a in the API formulation. Any overages in the manufacturing of the drug product, whether they appear in the final formula formulated product or not, should be justified considering the safety and efficacy of product. Information should be provided on the amount of overage. What is the information provided for the overage? That is the amount of overage, reasons for overage, why overage are added, that is compensate for expected or documented manufacturing losses. Then justification of the amount of overages. The overage should be included in amount of drug substance in batch formula. Then physiochemical and biological property. The physiochemical and biological property related to the safety, performance or manufacturability of the drug product should be identified and discussed. The physiochemical property that relevant to the safety, performance and manufacturability of drug product should be identified and discussed. This includes the physicochemical application of drug substance and formulation attributes. Studies could include, for example, the development of test respirable function of inhaler products. Similarly, information of supporting the selection of dissolution vs disintegration other means assure the drug release. The development of suitability chosen tests should be provided in the section C ICS 6 specification, that is a trace procedure and assessment criteria for new drug substance and new drug products chemical substances. Then decision ICSQ6B specification, this procedure and assessment criteria for biological and biotechnological product. Then discussion should be cross reference of the relevant stability data. Then manufacturing process development 2.3, the selection of the control, the improvement of the manufacturing process described in a 3.2 p 3.3 intended for commercial production batches should be explained. It is important to consider critical formulation attributes together with the available manufacturing process options in order to address the selection of manufacturing process, confirm the appropriateness of the components, appropriateness of the equipment used for the intended product should be designed, why equipment chosen for the manufacturing this intended purpose should be described. Then process development studies could be provided basis for the process improvement, process validation, continuous process verification, any process control requirements where appropriate. Each such study should address the microbiological as well as physical chemical attributes. The knowledge from the process development studies can be used as appropriate to justify the drug's product specification. The knowledge gained from the doing the development of the formulation provided the justify the finalization of the drug post specification. The manufacturing process and development program process improvement program should just identify the any critical process parameter that should be monitored or controlled. That is a granulation endpoint to ensure that the product gets desired quality. The process improvement program should justify the endpoint for the granulation endpoint to ensure the product of desired quality. For those products intended to be sterile and appropriate method of sterilization for drug product, manufacturing packaging material should be chosen as choice where justified. Significant difference between the manufacturing process should be produced batches of clinical trials, that is safety, efficacy, viability, bioequivalence of primary stability studies. The process described in should be discussed. The discussion should summarize the influence of differences on the performance, manufacturability, quality of the drug product. The information should be presented in a way of facilities comparison of the process and corresponding batch analysis information. The information should include the project name, the identity, that is batch number, use of batch as produced, that is the equivalent batch number, the manufacturing site, the batch size, any significant equipment differences. In order to provide flexibility of future process implement, when describing the development of manufacturing process, it is useful to describe measurement of systems that allow monitoring of critical attributes or process endpoints. Collection of process monitoring data during the development of manufacturing process can prove useful information to enhance the process identification. The process control strategy that provide process adjustment capabilities ensure control of critical should be described. An assessment of the capability of process to reliably produce a product of the intended quality, that is the performance of the manufacturing process under different operating conditions, different scale, different manufacturing requirements can be provided. An understanding of process robustness can be useful in the risk assessment of risk reduction. That is a quality SHQNS risk management glossary of the definition to support the proper future manufacturing process improvement, especially in conjunction with the risk management tools. That is a QC9. Thank you. Please and subscribe the channel.